graduate degree in statistics from the esteemed IIT Kanpur in 2018. So notably, he has been actively engaged in external outreach programs and has delivered his presentations at very, very prominent forums such as Fuse EU Connects, IACT, Conspec, and IACR conferences. In his current role, Soumitra finds fulfillment in contributing to the betterment of the human community through his work at Novo Nordisk. He strongly resonates with the company's purpose, which is to drive change in the fight against serious chronic disease. This mission is founded upon the organization's rich heritage in diabetes and is pursued through pioneering scientific breakthroughs, enhancing access to life saving medications, and actively working towards preventing and ultimately curing this disease they address. So, beyond his professional pursuits, Soumitra actively participates in discussions on equity markets and possesses a strong enthusiasm for embracing new technology. So, you can understand that what a wonderful person he is, and his work basically not only addresses the latest innovation from our academic background, but also it addresses very, very serious concerns in our societal, issues. So I would like to thank Professor Kaur, Soumitra Kaur, with us, and I will want to hand over today's session to you, sir, so that you can continue your session.
Thank you. Thank you, Professor Anuradha, for the nice introduction. And I will quickly jump onto the
Sir, I
presentation.
can see you, but maybe you are
Can you
your
hear me?
microphone is disabled. Just
Okay.
check once. I cannot hear you, sir.
No, he is or
Yes,
he
I
is
can
clearly
see you,
audible.
sir, but
I
I
cannot
cannot
hear
hear
you.
you.
Okay. Maybe I will just turn off the
Just
headphones.
a minute, sir.
Can
No,
you hear
it's
me
it's
now?
it's now it's audible, sir. Now it's audible. Yes, sir.
So now you all can hear me, right? I hope.
Yes, 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 sir.
Yes,
Yeah.
yes,
Okay.
yes.
Sure. I will quickly jump into the presentation. I have some network issues, so I will just turn off the camera. I hope that's fine. Sure. All.
Yes, sir. Your screen is visible.
Yeah. Thank you. So let me take you through my presentation today. So the topic is leveraging machine learning for predictive control in cardiovascular outcome trials and insights from imaging and wearable data. So I had actually given this presentation topic quite some time ago, and then I actually thought about. So let's think about like when I started to preparing on it, I thought it's not only cardiovascular outcome trials, but let's talk about more on the generic terms, like in other therapeutic areas, as well as I think most of you are not so much familiar with what in a clinical trial, like how a clinical trial is developed, or maybe like what I'm going to do today is tell you a story of a compound, like how a compound from a laboratory actually gets into the market, and then it actually saves thousands, like millions of lives. So let me take you through that kind of a journey, and without further ado, I will start the session. So what I am going to actually talk today is actually about second is yeah is about like to answer the question, can artificial intelligence, machine learning, and automation redefine the drug development? How we are actually harnessing the automation and open source innovation in terms of that. So now, after the session, you are also welcome to ask any questions or further contact me over the email, which I have in my first slide. But you are also welcome during the course also whenever you have any questions, you can ask me. So Before starting, a short disclaimer from me. So no data has been used from Novo Nordisk, and entirely it's for my simulated data or readily available packages from the R server of CRAN, and the presentation is only for education purposes. Now, I will start actually about limitations of current AI and ML technologies. For the past few days, you would have also heard about how AI and ML technologies are actually improving us in terms of in different areas. Now let us think about now ChatGPT is actually so much famous nowadays. So I just asked ChatGPT draw an image of clocks showing the time 8:30. So it can be 8:30, or even I have to actually checked actually on different times also. But then it actually shows the image as you can see here. Most of the images are showing 10:10. Why is that? The reason behind it is the most of the available images in if you Google it, show the images of clocks and all, most of them have the time as 10:10, and that's why it doesn't have so much of data to actually put in as a train in the training model, that it can actually generate times of different in times which is different from 10:10. So that's the reason why it cannot actually process it and show us the image which we wanted. Another example is I have just asked to draw an image of a person writing with left hand. That's also the same case. Most people are actually more than 95% people actually write with the right hand, and that's why it cannot generate a person writing with the left hand. So that's the reason there are certain places where current AI and ML technologies are not fully capable of, or not yet there to generate these kind of things. This is just an example. Now, as you have heard, I am from a clinical research background, we work on rare diseases. So what if there is not much data collected for a therapeutic area? Suppose a rare disease, an area which I am also working on is in a particular part of a CVD with some particular mutations in the heart. So then how will we actually solve those cases? So that's why it's of very much need that we all come together. So the core foundation of artificial intelligence and machine learning, as you all know, is statistics, mathematics, and computer science all together. That's the main part. But then, is there something beyond that? So you would have heard from people from aerospace engineering, and then also from different other fields, finance and all, on past few days. So that's why another important part here, which I would like to add on, is the domain knowledge, which is of very much importance. And today, I'm going to actually take you through the domain knowledge of clinical research. So that's our today's manual theme, that we will talk about how clinical research is actually jumbled into this core of statistics, mathematics, and computer science all together. So how it is actually helping us, the AI and ML is actually helping us in clinical research. But I have some things in mind because all of you are not so much familiar with the drug discovery method. So I will take you through four different parts. So in the very first, I will take you through the introduction of drug discovery, which will be very I have made it as a from a fundamental sense, so it will be easier for everyone to understand. And then I will take you through how we are developing through these new techs, and then I will give a simple example or use case with the support vector machines. And then the most important part today, which I will be talking about, is also like how we can actually, in clinical research, how we value data as much as molecule nowadays, which was not there back in maybe 20 years back or something like that, or maybe 10 years back also. But now we are valuing data as much as molecule. If your molecule works, but the data doesn't tell you the stories or the insights, then it's an issue. But how do we actually find the valuable information from data which actually comes exactly from the molecule itself? So now let me start. So this is actually a story of a molecule, and for you all to know, a drug actually takes around 8 to 12 years, according to the complexity of the disease, to come to the market. So it is a long journey that a compound is actually from the beginning of a lab, it actually is in the market, and then there is also post-marketing research. And the drug development timeline is actually divided into 5 steps in general. So the steps are, in the very first, it's actually discovery and development. What happens in this stage is, so there are suppose a new insight about the disease process that allows the researchers to design a product which will stop or reverse the effects of a disease. So suppose when COVID-19 came into the picture, so then this is a new disease, and it is an unmet need that it has to be some kind of vaccine has to be in the market. Then accordingly, there are thousands of compounds being tested in the lab in the very first. And that's where it actually starts with a preclinical research. So before testing the drug in the people, researchers must find out whether it has potential to cause any serious harm or any toxicity. And there are 2 types of preclinical research, which is like in vitro and in vivo. In vitro is, in Latin, it means like in glass. So then in the test tubes or the culture flask, the compounds are actually tested in the very beginning. And in vivo is like, which is in Latin, it is actually, the meaning is the living, within the living. So that means within animals, it will be tested in the very first. And then only it will go to the clinical research, which is the step 3. So step 3, it is the clinical research part where we biostatistician, or like where I also work mostly, which is the divided into mainly 3 phases of clinical trials. So phase 1, phase 2, and phase 3. So what is a phase 1? So phase 1 is like first human dose studies to evaluate the safety and understanding like whether the we can go on a further study. And this is actually having very less number of subjects, and accordingly different other topics also, other things also, features also about these studies. I'll talk a little bit one slide later. And the designs are mostly like dose escalation studies. So slowly you will increase the dose till you reach a certain type toxicity. And also it's not also only on toxicity, it's also about how much a body can take and how much level you can increase, so that it's also like you are actually reaching an optimal solution to your problem. So you are reaching the optimal solution that you are actually reversing the disease effect. And in the phase 2, it's mostly the dose response studies. So it shows like why the drug, or show the drug is effective or not, and describe the dose response relationship. So that's something similar to phase 1, but it's like a little bit more number of subjects on phase 2. So directly you should not move on to a larger sample of human samples, because then it will become, if there is any safety issue, you are actually having uh, some like so many people will be in danger. That's why you don't do it on a larger scale in the very beginning. And then in this particular phase 2 study, we decide on a dose which we will go on a phase 3 study, which is a much larger study with involving 500 to 1000 patients. And then in the phase 3, on those 500 to 1000 patients, you do confirmatory clinical studies to show that your drug is efficacious, and also along with you actually monitor if any adverse events is predominantly available. That means if the drug has any serious adverse events which is actually posing a threat to like make the product available in the market. Now, so this phase 2 studies actually gives like in the phase 2 we get the risk benefit thing, where we are getting the optimal dose, and in the step in the phase 3 we are getting it into the market, or like getting it into the efficacious probability. Coming to the regulatory review. So now from the phase 3 study, if we have an evidence, then the drug developer will actually put it under like FDA review. That FDA is like the regulatory from the United States of America. And then similarly, we have European Medical Agency, which is EMA, which is actually from the Europe. So actually you send it to your whole clinical domain package, which involves all the results of the clinical trial. Not only phase 3, it's also actually included phase 1, phase 2, phase 3 all together, and also the preclinical research. So you create something called as clinical study reports and then send it to them. So for further review. And then what actually it's done is like new drug application. We call it as NDA. And then FDA reviews it and then makes a decision to approve or not approve it. But here the story doesn't end. So then when the drug is already in the market, it's also important to look at what happens in real world. So whenever we are doing a RCT, which is randomized controlled trial, so randomized controlled trial is like every all the subjects or participants are actually blindedly actually assigned to one of the treatment arms. So suppose you have like a new drug, and then you want to actually test it on the humans. So you will actually randomly assign them to two different arms, and then you will test it after like suppose 1 year or 2 year. What is the benefit of that drug in terms of different parameters? So if you see the change to be statistically significant, then you can say the drug is effective. Also looking into the other aspects such as safety and else. So after the drug is in the market, despite rigorous steps which is involved in the drug development, there are limitations. So that's why the true picture of product safety actually involves over the months and years to make a product's lifetime in the marketplace. So what happens if you actually have any issue, if any person has an issue with a new drug, then they will actually report it to the regulatory authority. And also actually they report it to the company. And when it is actually reported to the companies, they are also like they also have to actually get it to the regulatory authority that these kind of questions we have received and these kind of feedbacks we have received. And then FDA will review the problems with the prescription and over the counter drugs, and they can also decide if any further causation is required with the dosage or usage information. Now, moving into drug development timeline. So it involves, as you can see, not only phase 1 to phase 3, I talked about preclinical test, but then also there is target validation, compound screening, and lead optimization. The most important part here you can see is we actually start with more than 10,000 candidates. And these candidates are nothing but each one is a molecule. So the molecules are different, but maybe like when you are doing 10,000 candidates, the molecules are not so much different, but then also it's different from different aspects, like according to the solubility and many other properties of a drug. So in the very beginning, we do something called as target validation, which is solubility and also stability. But apart from that, one thing is not mentioned here is something we do, which is called market research. So we have to look into how much the production will be required in this particular new compound, that we can actually have it parallelly with our ongoing studies, which is also very important. That means suppose a new drug or new like target we have found out, but then if we do this trial, then our like we all have the kind of like this production facilities very much limited, and nowadays it's a big challenge with some companies that we actually have limitations of how much we can actually produce, because the production of the drug has to be in a very high quality. And that's why it is important to look at the market benefit. So how much is the first question is there any other drug which is standard of care available in the market or not? If it is already available, how we can benefit from that particular product to our product? And then also like think about certain other scenarios, like whether our drug is will be on a IV like intravenous, or it will be a tablet. It's also important, if the drug is already available as a tablet, then we obviously will not go into a new drug which will be an IV. And then also the how much temperature control will be required for the drug, and then in how many days the medicine will expire. So we want to actually have dates of expiry quite longer, so whether the drug can be actually in the drug doesn't get deformed for a certain period of time. If it's actually very short period, then we don't want to go forward with it. So in these kind of scenarios, we need to look into. And apart from that, there is something called as competitive intelligence. So if there are other companies also who have started on actually dealing with the development of such particular compounds in that same field of class. So we call it like classes of medicine. Like you would have heard about medicines which are actually reducing body weight nowadays a lot, which is like Ozempic or Wegovy, which is from Novo Nordisk, and they are from the class of GLP-1s. So we always look at what other GLP-1s are actually being tested in the market, and then we want to try out like if that particular in that particular class, if we have competitiveness in the sense that we in the market is so big, so huge that we can actually reach the market within the time, keeping into our competitors also in mind, and then we can make a good business out of it. Then we actually think more about going forward with it. And then obviously there are other reasons, like in rare diseases, it's because of the unmet need why we do the clinical trials. It's not only for the business, but also for the unmet need, as I mentioned. And as you can see here in the very top, the cycle time is given for each of the phases. And as you can see, phase 1 is 1.5 years, phase 2 is 2.5 years, and phase 3 is also around 2.5 years to 3 years. It also depends on the therapeutic area. And you can see the probability of success actually increases from phase 1 to phase 2 to phase 3. And most of the cost for a new medicine or medicinal or drug development is on the phase 3, which is around 26% is on the phase 3. Yes. Now, one thing which is important here is we do not do a single trial of phase 1 or phase 2 or phase 3. We have to do multiple trials on each of the phases. And why is that? So, suppose the populations are actually different from one trials to another. You cannot actually combine different population in the same study, and there are several reasons for it. For an example, suppose you want to do a test of diabetes you are doing a new type 2 diabetes compound, and then it is a once weekly insulin, and you cannot obviously do it on a pregnant subjects. So that's why you have to do it as a separate study itself. And in the pregnant studies there can be different reasons or different thought process or endpoints, as we can say, the variables of interest which we want to look into. For an example, in a pregnancy study, what you would like to see is after the mothers actually give birth to the babies, so is the babies also diabetic? So, and in that sense, we also do some testing like the umbilical cord, which actually connects the mother with the child. Through the umbilical cord, is any of the drug particles going to the baby's body or not? So we do testing such like that after the birth. Okay, and then also generally for another example, so nowadays it's we actually Novo Nordisk were the pioneer of doing once weekly insulin. So in before it was only once daily insulins available in the market. Now we posed a question which is also available in some papers, and I was also involved in this clinical study. So the question was, it's a once weekly drug, and people can forget when they had taken their last drug. Now, what if a person actually wrongly takes like two medications within a week, or even three medications within a week, or maybe they maybe like it's a kind of a pen device. So you just press it and it actually goes into your body. So you press it on your hand, and the pen you press it in your hand, and it actually connects with you. It's a intramuscular injection. So suppose you forget, then what happens? So in 1 week, rather than taking once dose, you are taking twice or thrice the dose. So the doctors were very concerned on this. So we did a trial on that. So people were actually, it was a very small trial with like 30 subjects in each of the arms. So it was like I could take once dose and then twice dose and thrice dose, and then you are testing the results of different parameters and how they are different on it. And we saw very good results on that trial, and I have actually put a reference to the study results in the very end. Yeah. Now, all this time I was talking about the basics of like how we work, or how a drug actually develops. Then I would now go forward to like how we as biostatisticians actually contribute to this whole drug development process. Now the very first thing is how the projects are actually defined in this pharmaceutical industries. It's the projects are actually based on the therapeutic areas. So under a therapeutic area, suppose CVD, cardiovascular disease, there are also like several actually parts, like actually what cardiovascular diseases also have is a class of therapeutic area, but then under that also there will be different areas, or it is actually solving multiple questions or multiple problems. So so those each areas are also divided into such three parts like phase one, phase two, and phase three, and it's actually multiple trials are going on. And we biostatisticians generally get the role of trial statistician or a project statistician. So if multiple trials are going on, so each trial will have one trial statistician, and then the whole project will have a project statistician who will actually solve any issues in the project level. And then we have obviously clinical data scientists and trial programmers to solve any give us any insights or any further things. Now moving forward, the very first thing which we actually give input to is the clinical study design. So, and before moving even the clinical study design, we want to actually find out what is the clinical question of interest which medical and science department actually wants to answer. And this answer we are actually going to find out through RCTs or randomized controlled trials. So in that way, in that sense, what kind of study design we need to actually we need to actually go forward with. So that there we actually have to give inputs. But before going into that, maybe I will talk about a little bit of some of the things which you may not be actually fully aware of, which is like what is a placebo? So in clinical trials, often we do the test of in a parallel design, suppose like we do it like the active arm versus the placebo. Active arm is the new drug compound which we want to actually test on, and the placebo is nothing but a similar pill which is looking exactly similar to the active arm, but it's placebo. It has no effect. In terms of IV, it's like if you are giving an intravenous medicine, and then the other one, the placebo one, will be having the same color, same kind of look from the outside, but actually it will be saline. Basically, it will be just saline. And why we are doing it? To find out the placebo effect. The placebo effect means the patients should not feel that they are not getting treatment, or they should not get to know about that they are not getting the actual treatment. And but it is important to do that. I will talk about that, why we are doing it. And that's where the concept of blinding comes into the picture. So there are studies which are single blinded. That means the patient does not know whether they're getting a placebo or the active treatment. And then there is double blinded. Neither the doctor nor the patient knows whether the treatment is a placebo or an active drug. So these studies are actually done in this particular setting. So that's why it is called randomized control trial. So they do not know whether they are actually going into active arm or a placebo arm, because if the participants actually know that they are on a placebo arm, it can have an effect in terms of like the patient's health on a mental level also, that they know already that they are not getting the active treatment, so they will feel not even like there are questionnaires to solve like how they react to a particular drug, like whether they are actually feeling better or like worse from the beginning baseline. So obviously if you know you are receiving a placebo, you will say like I'm not feeling better, but maybe there is an effect of a drug when they're actually also taking placebo. So over time, maybe the degenerative process actually reverses itself, or it can actually the treatment can actually work in a worse way also. So to find out those kind of things, it's important to understand. Now here there is an example of a parallel study and also a crossover study. So parallel study means it's very simple. So you have the patients in in one of the arms, and which is randomly assigned. And then in crossover, what happens? So the sequence is, so you have still two drugs. One is active, one is a placebo. But then the sequence is different. So some subjects will receive the active arm first, and then they will move on to the second phase to the active to placebo, and then there will be some people who will be moving from placebo to active. And there is a in these kind of crossover studies there is a gap which is called a washout period. So it is there is like suppose 2 to 3 weeks of gap in between these two sequence of medicines, so that the drug actually gets out of the body, so whether like the previous drugs effects are not there in the body. Now, what is the benefit of one of each one of them? So, as you can understand, the parallel study will require more number of humans or more number of volunteers or participants in the clinical trial compared to the crossover study. So, the same like if you need 100 patients in parallel study, you will need 50 patients in crossover study, because then each of the 50 patients will be in one of the arms. And another thing is the time wise, you can think of the crossover study takes lesser number of samples, but the time is more because you need two durations, and also you have a washout period. But in parallel level, you need only, yeah, only one phase or one particular duration of a time. So when you have a phase 3 study, generally you do it as a parallel study because it's a huge population. And then to do the whole trial, and the trials are actually longer, maybe 2 years or 3 years. So if a trial is already 3 years, you don't want to make it like 3 cross 2 is like 6 years, and also adding a washout period, which will become 6.5 years. So you cannot stop for like wait for that much of period for the drug to come into the market or something like that. So that's the reason, like in the very beginning in phase 1 studies, we generally do a crossover study because it's a less number of participants and all. But then also there are other things like the rare disease, which I was talking about. In rare disease, because it's difficult to find out participants who will actually encounter with will take participation in the clinical trials. That's why it's mostly crossover designs also is actually performed on them. Other method or other clinical design. So there are certainly other clinical study designs. I will also talk about something called as umbrella trial. So umbrella trial is something like, so you have you have this same type of cancer, but then the cancer mutation is different. So it's happening in the lung only, suppose, but the mutations are different. So there are different mutations like rat, cras, and then also met. So these kind of different mutations are there. But then the drug is actually will be different in terms of according to each of the genetic mutations. And then that's actually called kind of like umbrella trial. And then the other trial design will be something like basket trial. So it may be different organs are actually affected through it, the kidney, lung, the it can be also different other things like, as you can see here, liver. So then the types of cancer is different, but then all of the cancer types has the same genetic mutation. So that's why you can have the same test drug being tested on all four of the areas. And accordingly, you can compare the results. So that's called a basket trial. Now, moving on to something else. What other things we actually contribute as biostatisticians? We have to, in the very beginning, write protocol along with other stakeholders, with medical and science, safety, and many other actually areas like. And what does a protocol actually contain? So protocol contains how a clinical trial will progress. So what kind of endpoints is of interest according to your clinical question of interest. So you have in the very beginning a clinical question of interest which you want to solve. Suppose in diabetes it will be how much of suppose you have a type 2 diabetes, and then the main endpoint of interest is HbA1c. So with a duration of like taking the drug for over 1 year, how much of HbA1c level you can decrease, and in what particular part of population you want to look into. So it can be you want to look into only pregnant women, as I mentioned, or you can actually look into adults with having some several other disease, like adults with having cardiovascular disease, because cardiovascular disease is very much related to having diabetes. So according to that, like your particular population, and then what is your endpoint of interest. It's not only the so it's one will be primary endpoint, and you can actually have multiple primary endpoints. So if you actually have a like a study of like CVD, then your endpoint of interest will be something like 6 minute walking test. So a person will be actually told to walk for 6 minutes, and their heart rate will be actually monitored over those next 6 minutes, and then it will be also noted down how much distance they covered on the 6 minutes. And then it has been actually proven as a biomarker. So in like after few, so then you will actually test after 1 year, so whether they can actually significantly walk more with keeping their heart rates stable after taking the drug, and it will actually compare between placebo arm and the active arm. And then we have to write something called a statistical analysis plan. So before even like the study starts, you have to plan what kind of statistical analysis you want to perform. And it's important to do it beforehand, because if you already have seen the data, though it's blinded, you can actually tweak the statistical analysis in favor of your study or in favor of your compound. And to remove that bias from ourselves, we have to actually define what kind of statistical analysis we want to perform. And then obviously there are several other like when the study starts, we do a lot of data checks, validations, and we send some queries to the sites. So for example, we have to actually check whether there is a subgroup behavior. That's what actually talks about from the data insights. So a particular subgroup is actually showing more effect or lesser effect compared to the other. So suppose in a particular Asian population, it's more prevalent to see a particular type of adverse events with a particular compound. Then we have to find it out. So those kind of insights we get from this drug. We get or we try to find it out. And then it was actually a story about this further indications, or what which we call it as post hoc analysis. So once the drug you have tested and it is actually successful, and you can see it's on a phase 3 study, it's a success. And then you see there is something happening with the drug, something happening with something else also happening with the drug. So there was this thing like one diabetic drug back in 2018, which is Victoza, which showed a benefit over body weight along with diabetic drugs. So generally, if you actually take a metformin or any other drug, you also reduce body weight for a certain period of time, but on a very smaller level. But this particular drug actually showed uh, around 5 to 10% of benefit of body weight. And then we actually tested it on a further on body weight. So this is very important to understand that we cannot actually say that Victoza or that particular drug actually reduces body weight from that particular study results, because that was not our clinical question of interest, and that is not how we actually started on the trial. So we have to be very much like our clinical trial results have to be very much in line with what was our initial question and what we wanted to solve. And that's accordingly have to be reported in the study results, and we have to interpret the results in that particular way. Now, once we have reported the results, there will be question and answers from the regulatory. For suppose there can be questions about, we can see the trend in the data. Suppose the regulatory comes with a question like we can see a trend in the data that a particular group of patients are actually having more AEs, adverse events of a particular type, maybe nausea or something else. And then we have to answer that in a way like look into the data in a close way and find out whether that result is actually significantly higher between the two groups or two like subgroups, as I mentioned. That's one part. And then we are also involved in something called as simulations. So one on a blinded study also, we can estimate the success of a clinical study. And generally, how do we do it? It's very simple. Suppose all the subjects are suppose 1,000 patients are actually assigned in a clinical study, and then they are actually into two of the arms, and then you are actually generating data on them, or you are actually collecting data on them for over 52 weeks, and each 4 week they come to the sites and gives their blood parameters and many other like tests, and we get the results. Now, before even all the subjects are reaching week 52, at week 24 or week 28, we want to actually test whether there is a significant benefit over the time, over this period of time. And how do we do it? We randomly assign, suppose ourselves programmatically, the subjects, each of the treatment arms, and then we actually simulate it. So it's just like a coin toss. If it's a kind of head, we do it obviously in the programming way. So if it's you can just say like if it's a head, then the subject is on treatment A, and if it's a tail, it's the subject is on like treatment B. And then you randomly assign it all these 1,000 subjects like 5,000 times, and then you actually rerun all the statistical analysis and then find out what percentage of times you are getting a success. So this is one of the way of doing it, but there are other ways also to do the simulations. So this is particularly one of some examples where we actually can actually get some insights about it. And then there are also other things like how we actually solve some questions from the sites. Like for an example, there can be anomalies in the data. So a data where the data is not in a proper not showing a proper trend. Suddenly there is a peak, or suddenly there is a the value, one of the parameter actually gone down. Then we have to look into, has there been any adverse events which actually was the reason behind this particular peak, or particularly for what reason the value went down. And then what kind of if there is any change over the drugs which they were actually taking parallelly. So there are some certainly like different therapeutic areas where you cannot test just placebo versus your drug, because there is already standard of care available in the market, and you cannot say that the subjects are not allowed to get the standard of care. So it can be like cancer. In cancer, they are we never do like something like a placebo versus the active arm, because the subjects need some kind of treatment, obviously. So it can be like chemotherapy versus a targeted therapy, or chemotherapy plus radiation versus a targeted therapy, like that. So there will be standard of care, and that's why statistics is very important. So here, how do you actually take into consideration that the effect of standard of care, which might be there in both the treatment arms, and then how do you test for your particular compound, what is the benefit out of it? Then you have to add it as a factor in the mixed model, and then analyze it separately. So I'm not going into much details on that, but there can be also many other use cases where we also give like inputs, something like recruitment time. So we want to go from a phase 2 to phase 3 study. Now we know what was the recruitment rate in a phase 2 study. So in a phase 3 study, if we want to actually complete the study within 3 years, in what rate we want to recruit the patients, and even if it is if it is feasible or not, that kind of estimation also we do. And some other examples is like the acquisitions. So when we have some acquisitions of other companies, like other smaller companies, then what we do is we generally test their data on like the validity, and whether the we do some simulations from there. Like generally the acquisitions are on after the phase 1 or phase 2 phase 2 compound generation. That means on a phase 3 time point, we actually get the data, we test the data. Now also another thing is because as you can see, like the sites are actually the major part here, that who actually recruit the patients. So it's also important to understand what is the particular number of patients we want to recruit, what like how many number of patients we want to recruit in a study from a particular site, because all sites may not be equipped with recruiting a lot of patients. And then we also want like heterogeneity in the population. You don't want like a study to be a phase 3 study to be only from one center, which is obvious. And generally we in a phase 3 study we do it from multi-center, so multiple centers will be there in multiple countries. And why is the reason behind that is, so regulatory authority, for example, Japan and China are actually do have this regulatory requirement that if any drug is actually approved in their market, there has to be like uh, quite a good amount of like volunteers from those two countries. Then only they will accept the drug to be marketed in their population in future. So what we do is, like, suppose there is a clinical trial of phase 3, you have total 1,000 participants. So 200 will be from US, 200 from India, and 200 from China, 200 from Japan. So in different areas of, and also 200 from Europe, from some other country. And then you will have, for each of these 200 will not be from one side, it will be multiple sites, so that we have a like good representation of the population. That's like the very beginning of our statistics, like how we learn statistics, that the sample has to be a good representative of the population. So that's why it's not only one site, it will be multiple sites and multiple countries, which from where we take the volunteers of the clinical trial, and then we test our drug on them. Now, another interesting topic is adaptive clinical trials. So what does it mean is, this has become also a very famous thing. So Suppose we have four compounds of a drug and then a control regimen. A control regimen is nothing but the placebo drug itself. So now you are testing the four drugs together along with the placebo. Then you have, like, the whole study is suppose of 3 years, and 1 year later you do something called as the first interim. So in the interim you actually test whether there is any significant like benefit over the beginning. And maybe it's not exactly significant benefit, it's more about it's not going in a worse situation. So for example here, novel regimen 2, as an example, might be what have happened is the particular endpoint which we are looking into of interest is actually not showing benefit, rather it's showing it's actually the patients are of in the control regimen is actually performing better compared to novel regimen 2. But it can also happen it's not only efficacy, maybe safety wise it's not optimal dose, it's not the dose, because of that particular dose maybe there are so many adverse events which is of serious concern, so they will drop it at that particular period. And similarly, you can see novel regimen 4 also is dropped, and also novel regimen 1 is also dropped. So in the dropped in the second interim. So at that point of time, suppose it's more like it's 1.5 year or 2 year later, you do it and you test it. And what is the benefit of it? So all the participants, all the regimens, if you are actually testing for 3 years, the total cost will be huge. And if you already have from the information from the interim analysis that the compound is not working out, then you will obviously would like to stop it, and also because of certain other reasons. So this is obviously another example would be like it's not always like four treatment, four different compounds versus the control. It can be also four different doses compared to the placebo. So you may be having a particular medicines 10 mg, 20 mg, 30, 40, and then you are actually testing it against the control arm. So you can actually parallelly test all the five arms together and find out which particular dose is actually the optimum. And then also it will you can reduce the cost by doing analyses in between, in the first interim and second interim. So, and also at that point of time you can actually improve your sample size in a better way because you have more information about the population at that point of time, which you can use from novel regimen 2 or novel regimen 1 in novel regimen 3, and then you can actually take it into a phase 3 study. So that's how, like, it's whenever the phase 2 study ends on, you can begin the phase 3 study with the same subjects also. That is also possibility. And there you can also use the information of novel regimen 1, 2, and 4. Yeah. Now I will take you through the next chapter where we will talk about the development through artificial intelligence, machine learning, automation, and open source programming. So in Novo Nordisk, how we are actually bringing innovation is through target discovery. As you have understood, like you have seen, there are 10,000 compounds in the beginning where we are starting with. So it's obviously not possible to culture them all in the same time by human persons. And so that's why the AI or machine learning is of very important, very much importance, that it can automatically understand that, and obviously with some particular setting and previous information, that what is the solubility we want, or what kind of how one compound is better than the other. So from 10,000 compounds to come into 250, it's not possible with human intervention. So it will be done by different machines, which is actually available in the labs. And then you will do something called as uh, so here the robotics actually is used to do that. And then you have knowledge mining in terms of like when you very much start on a target, so what particular target is of more importance, so where we are actually not have not met the need of the human generation. So that's one question we want to actually answer. And then through literature mining we can actually solve it, in the sense that you have to for a particular therapeutic area, look into all the papers and publications or whatever information is available in the market, and try to get some information or some prior information on that, so that we can use it in our future trials or future innovations. So that's how it is done. And then also there are something like how we can optimize the sites or the trial designs, which I talked about a little bit on that, like how many particular number of subjects we should actually have in a clinical trial for that particular from that particular sites. And how do you know that? You have done many clinical trials on that from that particular site before, and you know how much of recruitment is done at that particular site, like what is the rate of getting people from there, and then what is the dropout rates, which is also very important. So subjects can drop out from clinical trial anytime they want, and there can be several reasons for it. So maybe the drug is not performing well, maybe it's like because of their family doesn't want. Yeah, we do get some reasons of that. The family doesn't want them to actually be in the clinical trial and all. And then also we use it for pattern recognition in event adjudication. Just to give a hint of what is an event adjudication, suppose a subject is actually hospitalized, and then we actually do not know that whether it's a cardiovascular hospitalization or non-cardiovascular hospitalization. Maybe the investigator or the doctor has mentioned it as a cardiovascular hospitalization, but the underlying disease was different, which was found after uh, different clinical tests. So suppose after MRI or some other like PET scan, it was found out it was actually cancer, which was the prevalent reason or main reason of that hospitalization, which actually will not make it as a cardiovascular hospitalization. It will become a non-cardiovascular hospitalization because the main reason of it was cancer in the background, which was not found in the before. And then there are other things like deep learning in product inspections, AI augmented process analytics, resource allocation forecasting, patient journey predictions, and targeted marketing material. Yeah, targeted marketing material, we already know a lot that nowadays it happens a lot that we know that who which participants are actually having. We have a huge database of which participants are actually have in before has taken our drug. So in that sense, we know the target, like whom we have to reach out if a new drug is available in the market, which can better their future or their life. Also to mention, we also have our own Gen AI tools and ChatGPT, as well as AI assisted job advertisement generators. Now, found data is something else which we want to say. So it's actually how we can democratize data across Novo Nordisk value chain. So there are two types of actually data, but one is primary use, one is secondary use. Primary use is like why the data was actually collected in the very beginning. It was actually the clinical trial, which is under the consideration why we have collected the data. But then can that data give us information for any future studies or future recruitment or future any other indications? There are certainly many reasons for it. So then if we actually use it for any secondary purpose which is not mentioned in the protocol or which we didn't know, we just got to know from the inside of the data, that's actually called secondary use. So these are easy to connect. It can multiple data types and sources. I will also talk about omics today, which is actually our proteomics, genomics, and all. And that it's easier to solve in advanced clinical data insights because it gives a lot of data, lot of information about the patients. And then with this informations, we can also reach something called as translation research, which is like personalized medicine, which is very interesting to learn about nowadays. Uh, I talked about something called like literature mining, target selection. So these are like predictive modeling. These are done at the drug research level. And then from drug research, when we go into drug development, in between, there is something as biomarker development, and then also the data insights and early detection. In drug development, also there are like site analytics, patient recruitment, reduction of dropout. So how we can actually reduce the dropout by selecting sites which will perform better. And then in clinical study submission also we use it. I have a very good topic about that clinical study submission, how we use it in Novo Nordisk. I will actually talk about that and also about wearable device and study simulation I have already discussed on. For particularly, I would talk about a little bit on the biomarker development. So what is a biomarker? So biomarker is a kind of like the endpoint of interest which actually shows there is a benefit for a particular therapeutic area in the population. For example, for a cardiovascular disease, it's like the high sensitive CRP protein. And then also there is something called NT-proBNP, 6-minute walking test also I talked about. There is also lipid profiles, so which actually talks about whether a person's lipid profile, if it is actually showing a particular trend, then it can actually interpret whether a person can have a CVD, a cardiovascular disease in future or not. And then also in diabetes, it's mostly the HbA1c, which is 3-month average of blood glucose level. So it tells you in the past 3 months whether you must have heard of this HbA1c test. So it tells you that in the past 3 months, on an average, whether your blood glucose fluctuated a lot or not, and then it actually reached a certain period, certain mark or not. So anything above 6.5, which means like you are highly diabetic, and anything between 5.5 to 6.5, HbA1c is in percentage, is you mean like you are pre-diabetic, and below 5.5 is good, you are non-diabetic. Okay. In cancer, what happens for biomarkers is mostly nowadays it's the prevalence of a particular protein or a gene. And what we do here is like profiling of the mutations. And for cancer, it's also like you would have heard about prostate cancers, and the type of test which is done for as a biomarker is like prostate specific antigen PSA test. And then there is also PDL1 test, which also tells about a particular protein level. If it has gone quite up, then you have then someone has cancer. And then how you can treat it, it also depends. There are targeted drugs which actually targets those PDL1 compounds. For kidney, it's mostly serum creatinine, which is tested, and also like urine to albumin in urine, albumin to creatinine ratios. So these are the type of biomarkers. Then how do we develop them? That's the main question I want to talk about. So biomarkers are developed in a way like you have when we do in the very first a clinical trial, you have many different parameters to test in a phase 1 or phase 2 study. Then you look into in different questionnaires of questionnaires. In terms of questionnaire, what I mean is that the participants have to answer some like multiple choice, like some questionnaire, in the sense that how you are feeling after taking the drug for 1 year. So are you feeling better or worse or something like that? And then you find a threshold in different parameters that if someone is actually having 1 level improvement or 2 level improvement, what is the average change for a particular endpoint for those subjects? And then from that you can get a threshold that if there is a like 50-meter increase in the 6-minute walking test in the subjects who are having 1 level of improvement, then you can say that that's the that 1 level of improvement with 50 meters means so 50 because it shows from that particular biomarker, it shows from that particular endpoint that you can say if this particular endpoint has shown an increase, then the participants are significantly performing better in terms of their quality of life. And it can be also seen in different in relationship to the other endpoints, such as the I was talking about CVD. So high sensitive CRP protein or lipid profiles are also becoming better for those subjects who are actually having 1 point improvement compared to those who have lesser than 1 point improvement. So in that way, you can find out different biomarkers. Coming to the other thing, which is very interesting, is the imaging in clinical trials. So nowadays, imaging is of very, very great importance, because what you do is like generally how a test is done. So generally you go on so suppose someone has like a disease, you go on a CT, MRI, or PET scan or SPECT CT, and then the doctor actually looks at the results or the MRI or the PET scans reports, and then find out whether there is a particular lesion which is of interest to them, and whether it's malignant or like benign. And then they find it out what type of cancer and all, and for that you need again a biopsy and all. So identifying tumor lesions, type of cancer, as well as the stage, whether it's the first stage, second stage, or third stage, can we do it using artificial intelligence? Yes, it's there has been a lot of work being done on that, because Nowadays cancer is becoming very a lot of people is actually suffering from cancer, but then it's important to identify them at a much faster pace, and also in a better way, or correctly to finding out. And that's where actually the identifying the tumor lessons in particular different parts of your of our body is also very important. And that can there actually AI can actually help us a lot, because you feed in like data of lakhs of people, which are like for example, in the Tata Cancer Institute also has this research program ongoing, and they have several papers on that. So they feed into their model the PET scans of like all the patients in past 10 years, and now they could actually test it on the patients, on the current patients, CT scan or MRI or PET scan, and then find out whether there are tumor lesions. And they have not still found out the type of cancer as well as the stage, and that's the next stage. That's where the mutations will actually come into the picture to look into that. Another thing is, you'd have known heard of something called as biopsies. So biopsies means like a particular lesson from the body has to be taken out, and it's an invasive procedure. And uh, and sometimes what happens, so after the disease has whether the disease has progressed from the initial level, people, the participants or patients may need to undergo through this invasive procedure of repeat biopsies. And as you know, the cancer patients are always already very ill, and doing again an invasive procedure, it makes it a lot harder for them. So can we use the only imaging to find out whether the mutation of in their body has changed from the beginning to at a later stage? So this actually the imaging actually helps us to reduce the need of repeat biopsies. And that's very important. That's one part. And then also another thing, the insurance authorization. Can this all happen at the same time with the use of AI and ML? So you have the scan. So in the very first, as you can see here in the side, in the side that you have an exam order, you schedule it, the insurance authorizes it, and the scan is acquisition, and then you interpret and report and communicate the results. So everything can happen in the seamless way when we have AI and ML into the picture. So everything can be faster in terms of with the use of AI. Another thing is, nowadays you would have heard of different stories of digital health technology, something like the wearable devices, continuous glucose monitoring machines, and hospital registries, as well as insurance data. So these all are called something called as real world data. So in real world setup, how data from these devices you can use to train our model and find out data on a real time basis, and find out whether it can actually we can benefit from that. So rather than waiting for a certain period of time, can we find a trend in the heart rates that will actually find out that a person is more prone towards heart disease in future? And also there is like SpO2 levels and other things which actually nowadays it can capture. And probably you will also know there are like nowadays the like smart watches which can also give you ECG reports. It's not very accurate, but it is trying to do so. And think about hospital registries. So you have from the hospital registries the data on what was the medical history of that participant. So wherever they have gone in the beginning of life to the end, to when they are actually like maybe a person is actually hospitalized at the age of 55 with some disease, but then to understand what he had at the beginning of their life. Suppose he had some kind of operation at the age of 17. So how you can actually gather all those information on a personal level? So that's what like personalized medicine. So what could be the best treatment can be found out with knowing the whole medical history of that person. And that can be also only done through this source, because it's very difficult to find out a data which is around 30 years old, but it can be easily done with a digital health technology or other data sources such as the hospital registries and insurance data. And another good thing about this is like we call it as decentralized clinical trials. So the subjects do not have to go to the sites, and actually from their home only, with we will get the data through their wearable device and continuous glucose monitoring machine, and we can find out whether the drug is actively working or not. So they do not have to go to the sites, and it will be much more easier and they will be flexible on doing so. And then there is also omics data, which focuses on the molecular part of the story. So it actually is like the genomics, transcriptomics, proteomics, metabolics, and other things. For example, I'm not going into much details on this, and these slides will be shared with you. So for cancer, it's mostly transcriptomics and epigenomics, which is more prevalent in terms of omics. So generally, the transcriptomics is actually the RNA, which is like which tells us which genes are turned on or off. So mostly it gives the gene expression path. And when you look into epigenomics, it actually shows the change in active genes. So there is no change in the DNA sequence, but there will be change in the gene's activeness or modifications. So this is how you actually find out which particular domains or which particular information is more important to find out through omics or different spatial and temporal character of the data. And then we have something called as digital twins. Digital twins is nothing but so you have a patient, and then for the participant, you can have a digital twin, which is something like you have clinical trials data from earlier trials. Then you don't need to actually have a participant from your control arm. You can actually generate the control arm's results through the data from previous trials with taking the prior information from other studies. So maybe like the patient who is actually, you have a patient with an age of 23 who is a female, location was in US, weight was 60, heart rate was 100, blood pressure at baseline was 100, like this. So this is a actual participant who is taking the active treatment arm. Can you find out with the previous trials, like every pharma company has done a lot of trials on the placebo, a lot of trials, and they have a lot of data on these placebo subjects. So can you find a participants with such baseline characteristics and make it as a digital twin of this participant and simulate the data, which is at particular these like 4 week difference, what will be the compound's value if they're actually not taking any treatment or drug? There will be some amount of fluctuations. It's not that they are always same, and they are never same actually. Over the time the values will change, but with respect to these particular characteristics, what will be their value? Then what will happen? You will have still a parallel study, but then you are actually not doing the, like, you are recruiting, not recruiting the patients, so you are reducing the burden of recruiting more patients, rather using it as a digital twin. Then I was talking about something like clinical study reports. So every time you have done a clinical study, you will have something called as clinical study reports. In the past, it was actually 12 weeks, like 3 months would have taken to generate a clinical study report after the study ends. So which actually summarizes all the clinical study results, as well as gives us all the like interpretation and everything, any further like insights to the data and all. Now, with this structured authorizing tool in Novo Nordisk, we can make it from 12 weeks to just 10 minutes. And how we do that? I have written something else also, but I do not have much time. I have 20 minutes more. So I will just a little bit jump on to further. So here you can see something from the CSR clinical study report. As you can see here, it actually is also available in the market and web. I have given also the reference, so you can go and ahead and look into that. So these values, that the total decrease in HbA1c was observed with the largest total decrease in HbA1c was observed with Adaglida and
Sorry,
mean.
sir, the picture is not visible. It's not clearly visible.
Okay, but I will just say what it is actually there. So the mean HbA1c at baseline was around 8.3. The level here is 8.3 percentage. So where these values are actually coming from? This is actually, so we have in the back end, we have the, like, these tables are populated. So these values are actually coming directly from that particular folder, that particular file, that it actually generates this value, and then it is actually automatically taken into here. So previously, people here, there are medical writers who will write it like manually, and it can also involve mistakes. And now it automatically comes from the programmatically generated outputs directly to the CSR, clinical study report, and it actually puts the images as well as other things. The more about this process you will find in the reference here. And I'm sorry the image is not so clear, but you can go ahead and look into it later point of time also. And then we also have our own version of ChatGPT, which is also like ChatGPT 4.0, and we also have DALI, which is the image generation. But then how it can actually help us? So obviously we are like from statistics background. We don't have like research literature idea about the medical compounds. So for a particular question, which has been asked by medical specialist, how do we answer that? We summarize the literatures which is available about the medicines in simple terms using ChatGPT, and then also we can get a lot of support in statistical programming, like we use generally R and SAS, and we get a lot of support, especially in R, from ChatGPT. And then we have a problem in hand. How do we solve it? It's generally obviously like when it is a statistical problem, it's more of it's us who are solving it. But suppose you want to actually look into what can ChatGPT help us into, like how we solve it in a different way, slightly different approach. So it actually don't give like it's not the solution, always the solution giver. So I will talk it we will speak about it as a more of a companion of you, who is actually helping you out in giving you new insights or new ideas on how you can use it in future. And then how do some simple things I would like to say, like how we have actually developed R in Novo Nordisk. We use different internal packages to do plots and tables, and exporting the tables into our folders and making it as a kind of full PDF with all the reports. And for that, we use R. For an example, we have like different sections in our we have a website actually. We call it as output catalog. So you can, as you can see, there are different sections which we actually generate for the CSR, clinical study reports. Like for, and the very first is here, you can see 14.1, which is like demographic data. So what was the baseline characteristics of the subjects at the baseline? And then the statistical analysis is mostly in the 14.2, which is like the efficacy data. And then you have several tables of safety and other things. To show you an example, we have something like here, as you can see, adverse events. So you have two treatment arms, Mars and Twix. These are actually chocolates, and this is a like draft output. So we actually check what was the total exposure in terms of years that for each of the treatment arms, and then how many number of events were actually captured, and how many were serious or non-serious. This is a pretty basic one. But then you have such similar to this, you have multiple summaries which you want to actually create. And then you have the outputs as well as the code examples side by side. This is obviously internal thing, so we don't have it as a reference link or something, but we do it in such a way. And our main focus is in future, this catalog will make it in a single click with the understanding the data from the clinical trial will generate all the outputs by itself, rather than we getting involved and run it manually for each of the trials. And for that, what is most important is all the data sets or all the backend data has to be in the same format. Now, I will quickly move into our next chapter, chapter 3, which is the use case of support vector machines. And I'm sorry, I will be a little fast now from now because we have only 14 minutes left. And to give you a background, so let us start with a two-dimensional data. It's having three variables, but suppose we have height and weight of a lot of patients, and then we actually, what we do is we do a scatter plot of them. So in the x-axis, it will be x1, and the y-axis, you will have x2. So height and body weights. And we want to find out the Z is a categorical variable which tells whether they're in active treatment arm or they're in zero, means like they're in placebo. And we want to actually find out how, if we get in future a new height and new weight, can we classify it's actually not active or placebo. I'm sorry. It's just like some other category. So suppose like some other particular category, which actually defines them in what class they actually belong to. So you want to actually have define a new height and weight. If this pair, if you have, then what will be the color of it, or what will be the class of it, whether it will be in the red class or in the green class. So for that, you will actually train it on these kind of heights and weights, just like any other like machine learning models. So you'll have the training model, you will have a lot of, just to example, for an example, you want to actually test if I give you a new apple, will it be something in the red line or will it be in the green side? So you want to actually well, like define or classify them in an easier way. So for that, what you do, you actually train them on a lot of images of apples and a lot of images of oranges, and then you train it. So this is particularly a problem of classification. So mainly, what is the solution or goal we want to find out? We want to find out the particular decision boundary, which will tell us in one side of it, it's always apples, and in the other side, it will be oranges. But then there are a lot of different options, as you can see here. But we want to find out the best solution, the one which is actually giving us the optimal line, which is separating this class, the whole space into two different classes. In that sense, support vector machine becomes very, very important and interesting also. And the reason behind it is there are only 2 points which are used to find out this particular like optimal line, which is the most extreme cases. As you can see here, maybe I can make it. As you can see here, the two extreme points, which are actually called support vectors, because in 2 dimension, it's just like x1 comma x2. But then when it's like multi-dimension, then it will be a point which is having more than like two dimensions. So it will be x1 comma x2 comma x3 dot dot dot. It will be just a vector. That's why they are called vectors. So these just two support vectors are the ones which is actually defining the maximum margin, this whole margin. And if it is, obviously you can understand, like it's more than two dimensions, then it will not remain a straight line or just a line. It will become a hyperplane. So how this line is actually like, how do we find it out? So we find it out by finding the equidistant distance from these 2 points. So this particular line has to be equidistant from these T2 points, as well as the sum of the distance has to be, like the perpendicular sum of the distance has to be the maximum. So with these two definitions or two criteria, we have to find the maximum margin line. Now, why we are doing it? How this SVM is different from the others? The reason is, again, this only based on these support vectors, we are defining this maximum margin. So how it helps is, suppose you have a apple which is very much looking like orange, but then where you will actually define it, you like where you will actually classify it. So that's why we are taking the most extreme values so that we can classify it in more extreme cases when there is a possibility of getting such answers or such results. Now, it was one thing I should actually mention here, that here you can see the data was linearly separable, that you can simply actually draw a single line and you can find out the answer. But then what if it is not? So what if the data is not linearly separable? What you will do? One solution which I am going to talk about is increasing dimensionality. Let us take a simple example. Suppose this is your fx function on a 1 dimension, and these are the 9 points, as you can see. Now, I will subtract 5 from each of them. And then you can see the 3 red points are in this side, the 3 other red points in the other side, and 3 green also in the other side. I have 0. Now I squared them. So it will actually obviously make a parabola, and then you can have the linear separator. So what you can understand here, from increasing the dimensionality, from going into from 1 dimension to 2 dimension, you have a linear separator. So that's how the mapping from a 2D space you can go to a 3-dimensional space, as you can see here. which will actually give you a hyperplane which will define the two classes as you need. So first you have the mapping function, and from mapping function, you will again do a projection of it. And how do we do it? Actually, the underlying mathematical concept is Lagrange's method for constraint optimization, which helps us to go from to find out the exact right mapping function from a two-dimensional space to a three-dimensional space, and then again project back to the two-dimensional space. Okay, this is one way of doing it. But then you can understand from going into or from adding an extra dimension, it means a lot of complexity when we are talking about not only 2D or 3D, but multiple other dimensions. So mapping into higher dimension can be highly compute intensive. So what is the other approach or solution? That's the Gaussian radial basis function, which I am going to talk about. It's a kernel trick. It's something similar, but it's very interesting to know about. So, and it's very much similar to the like you will see this this particular element in like normal distribution also. So what happens this for each of the points, you will actually find out the distance of the point from a point which is landmark, and this landmark is nothing but 0, 0. And suppose we take 2 points which is near to landmark and which is further from landmark. So what will happen here is, so if the point, suppose this one x, is near to L or the landmark, then what will happen? It will tend towards 0. And then e to the power minus something 0 or something like very small will be very near to 1. And with this particular value of 1, it actually increases the height of the element, and that's where the value of the kernel actually is the vertical distance from that particular landmark, and that's how you get the density. Similarly, if a point is much further from further from the landmark, then obviously you will have e to the power minus some larger value which is tending towards 0. So that means it will be outside in these kind of areas. And then how do you do it? So this circumference is actually like now this base is actually the now it's making a kind of like you can draw a circumference around it, and then according to the circumference you can again go back to the two-dimensional space and find out a particular non-linear curvature which will actually define the two classes. So inside the circumference, it's any value is obviously greater than 0, they will be green, and outside the circumference it will be close to 0, which is red. So the aim here is to find out the right sigma, which will define the two treatment arms. Two classes. Sorry. Now moving into what if it's not so simple? Then you have more than one radial basis functions. So you have like the very first one. Suppose KXL1 will actually cover for this particular part, and KL2 will actually cover this part. And then whenever the values are actually greater than 0, you add up the two values, then it will be green, otherwise it will be red. And there's a very good book, Hasty Tributary of Elements of Statistical Learning, which actually talks about these tricks. Now, I have a problem statement, which actually is, just quickly go into it. So suppose you have a data of quite huge data of like from National Institute of Diabetes, Digestive and Kidney Disease, and the objective is to diagnostically predict whether or not the patient has diabetes or not. So the aim is to train a model with SVM algorithm based on the training set and check whether the model performs well in predicting the response variable to predict whether the person has a risk of having diabetes or not. So the whole data set now is divided into two sets, like 75% is like training and 25% is test with the unique IDs. And we will remove the response variable because it is already there, but we want to remove it so that it can actually from the test data set so that it can actually estimate or predict it. So we will actually now train the support vector machine using the radial basis function, and then we will test it on we will test the algorithm on the test data set to check the confusion matrix. You can look at it in R. The function is SVM, and it comes from E1071 package, which is also available in the CRAN. I will directly jump into the result. So we had 192 set of outcomes, and then uh, one fourth of the test set outcomes. You can understand like four times of this data was used to train the model, which is around 760. So 768 outcomes were actually tested, or was having the whole data set was having 768 data points, and then a 192, one fourth of it has been actually tested after training it on the rest of the training set. And the model actually has predicted 154. Which 154 are those? Like whoever was observed was actually zero, and then it was predicted also was zero. So that is 110, and then similarly 44 is the correct one. So the 1, 1, and 0, 0. This is the confusion matrix. So 154 out of 192, which is around 85% of, I think 80% will be actually predicted in a good accuracy. And so what happens when you actually increase the percentage of training set? So obviously it will increase or make the model better and better, and it will increase the model's accuracy. And then also you can use something called as cross validation and parameter tuning to enhance the model's performance. So it is also not only the sigma you can tune in, you have many other parameters. I just took the very simplest model. Now I will move on to the value data as much as molecule part. Not go into this because of time constraint. Okay, so as a statistician, we all want the sample size to be as least as possible, because we want to actually spend less amount of time and as well as power, as less amount of time, and obviously the cost will be also more and more when your sample size increases. But we want to also have a very good amount of statistical power. Statistical power is not is something like whether your test actually can claim, or like maybe I can go a little bit before, yeah, which actually talks about it. So the power actually refers to the probability that the study will detect a true difference or true effect if there is actually one existing. So it's like the ability of the study to reject the null hypothesis when it is false. And generally we take it as a percentage, which is like more than 80% or so. So whether you can reject the null hypothesis when it is actually false, that's what you want to test. But you want to actually increase obviously more and more that. But then slowly, we are going to in another direction, which is of more importance, which we should be caring about, is considering efficacy of the drug along with the cost. So not only be cost effective, but whether our drug is actually extending beyond the initial occurrence. So whether the efficacy is actually extending beyond the initial occurrence. So are we providing persistent effect or consistent benefits in preventing or managing the subsequent events? So that's why it brings us to the question about we need to move from our comfort zone and consider the cost, and hence look for persistent effect and look through the patient's journey. But then how and why? So for an example, when we are in a CVD disease, we look into not only the first event, but also recurrent events. So any future events which is occurring. So this is an example of when a subject is not given any medications. So they have like 3 hospitalization and then a death at a certain point. In the first scenario, when the first drug is given, first set of drug is given, it prevents all the events equally, as you can see, and there is no death, which is good. But then look into scenario 2. As you can see, later events are delayed. So that means the drug takes time to be effective. So not only the very first occurrence actually tells you the information, but also the recurrent events. So that's why you have to look into the patient history so much. For the last question, last part, it's very important, which shows the very first event is actually delayed. But does it mean the drug works? But then you can see the next 2 events happens very close to each other, very close to the first event also. That means it develops
So,
resistance at a later point of time.
sir,
Yeah. Yes.
from this coordinator side, actually, I want to tell you that we need to wrap up this session because we have online feedback session and also the valedictory session from 6:30 p.m. onwards. So if you
Okay.
please kindly wrap up the session.
Yeah, I will do it in 1 or 2 minutes then. Is it fine?
Yes, sir. Yes, sir.
Yeah.
Absolutely
Okay.
fine.
Thank you. Now, how we look into the patient's journey throughout the study is through this is like something from the test data. We have something called a shiny tool which looks into different parameters throughout the study, how the different parameters like lab, vital signs, adverse events, medical histories actually is also in any of the subgroups or any of the particular sites or countries. And then we actually look into if there is any insight from the data. For an example, here I am choosing a particular SOC. SOC means nothing but like a the standard of care. So the subjects who are actually taking the standard of care and not having the standard of care, for them, what kind of different demographics we have and different other parameters. And then you have like for cardiovascular disease area, you have different classes. So class 1 to class 2. So whenever the class increases, then it's worse. And when the class decreases, it's actually better. So it actually shows for a particular, like you have filtering conditions of whether the subjects were taking any pacemakers, whether they were smoker, they were using the standard of care or not. And then you can see how the patient's journey changed from time to time. These are the references. And I have also given some publications where I am also one of the contributor, but it was interesting to read about these things. In the very end, I will tell something from the Grandmaster Gary Kasparov. So our humanity is not defined with any skills like swinging a hammer or even playing chess. Machines have calculation, we have understanding, machines have instructions, and we have purpose. And machines have objectivity, we have passion. We still need to get the help of a new intelligent machines to turn our grandest dreams into reality. So let us all dream big. Thank you. That's all I had
Thank you.
for today.
Thank you so much, sir. It is really, really a grand lecture. What I can say that it brings us immense joy to hear from you, sir, because it is my immense gratitude to express my heartfelt thanks to you for your enlightening and impactful session. So I would first like to ask my dear participants if anyone has any queries, then feel free to ask Dr. Kaur for any kind of concern or any kind of collaboration if you want to do, or any kind of research works which Dr. Kaur has undergone or underperformed. If you want to be associated or if you want to get to know something more, then please, dear participants, you can raise your questions. Yes, Dr. Kaur has already given his email ID as well. So, my dear participants, if you have any queries, then you can ask. Okay. So, Dr. Kaur, I think actually, the participants are very much, excited too, because it is a kind of, 6 days ATAL FDP program that everyone is going to attend. And also they have some online test and valedictory session as well. So our time is very kind of, bit tight. So without wasting a time, I just want to tell you that your expert discussion on drug development and the vital contribution of biostatistics has been truly enriching, and we have learned that drug development is a very complex and multifaceted journey that requires not only cutting edge scientific knowledge but also careful planning and data analysis as well. So, your deep understanding of the scientific and regulatory landscapes has expanded our understanding of the rigorous process which actually involved in drug discovery and approval. So, equally important, you also highlighted the indispensable role of biostatisticians in ensuring the success of drug development as well. So, your expertise, sir, in data analysis, clinical trials, and statistical modeling is critical in making informed decisions that ultimately improve patient outcomes. So, your insights into how biostatistics can optimize trial designs, minimize risk, and ensure efficacy and safety were thought provoking and deeply insightful. So, on behalf of all the participants, I extend our sincere thanks to you, Dr. Kaur, for sharing your invaluable knowledge and experience with us. So, your contributions not only deepened our understanding of the drug development process, but also underscored the critical role of biostatistics in modern healthcare. So, thank you once again, and
Thank you.
thank you very much.
Thank you so much. It was a very nice experience.
Yes, and Dr. Kaur, I'm just going to take 2 to 3 minutes from you so that I can request all the dear participants to please switch on your camera so that we can take the attendance of the last session of this 6-day auto AICT online FTP. Just give me 1 or 2 minutes, dear participants. Thank you. Thank you so much. Thank you, sir,
Thank you.
for your time and effort. Thank you so much. We will look forward to collaborate with you in future as well. Thank you so much.